Hello, everyone. Welcome to Classics in Neuroscience, Week 11 interactive session. I am Ramya Shekhar. I am a PMRF TA for this course. Is my voice audible and is my screen visible? Okay, so let me first begin by discussing a little bit about the two Nobel laureates who were discussed in this week's sessions. So that is Bernard Katz and Roger Sperry. So let me first briefly recap what we have learned about these two scientists, and then we'll go to the previous year assignment questions and discuss them in greater detail. So first, starting with Sir Bernard Katz. So he was knighted. So this is Bernard Katz, who like we will discuss about his personal background and the relevance of his personal background to his career and his contributions and the challenges that he faced in his life and the his role in various aspects such as in understanding the nature of neurotransmission at the neuromuscular junction, his role in understanding various other aspects, his contribution to the Goldman Hodgkin Katz equation, and then just a minute. Yeah. So his contribution to various different aspects of neuroscience, his leadership, and his legacy. So, who was Bernard Katz? He was born to a fur trader in uh, at that time in Germany, but at that time he was a Russian basically. So he was a Russian Jew. So at that time there was basically at that time there was like war was happening, right? And so initially he was at a place where there was Russian hegemony, but then what happened was because of the Russian revolution he became basically stateless. So he was born in Leipzig, Germany, and but because of all of these political and these kind of problems, he basically became stateless until the age of 30. So he studied in Germany, but at that time, what happened was there was a lot of anti-Semitism or anti-Jewish sentiments were going on at that time. So at the time, just the Nazi regime was basically slowly taking up more and more power. So there was a lot of difficulties that he faced because of that, even during his school life, even during his later stages in his career when he was trying to publish papers and the journals would not let him accept those papers because he was a Jew, and so his some of his like articles were declined because of his ethnicity, and he also had to basically so he had to flee from Germany, and he took refuge in the United Kingdom. So he had to take something known as the Nansen Pass. So because he was stateless, he was issued the Nansen Pass so that he could come to the United Kingdom and continue his studies and work basically. So he, after his completing his MD at the University of Leipzig in Germany, he like because of the circumstances, apparently he even initially considered migrating to Palestine, but then he was discouraged from doing so by the one of the Zionist leaders at that time, and then he was given a recommendation. So at that time, because of some of his research, one or two articles that he published, he also came to the notice of A.V. Hill. So A.V. Hill was a notable researcher in in London, UK. So he came to the notice of A.B. Hill. Also, his one of his professors gave him a letter of recommendation. So his one of his professors, Martin Gildemeister, also gave him a letter of recommendation so that so that he could join A.V. Hill's laboratory in London for his PhD. So finally, in 1935, he managed to escape from Germany and come to London for his PhD with A.V. Hill. And he regarded regarded A.V. Hill as a very, very important person in his life. A.V. Hill was his mentor, and A.V. Hill also helped him in various aspects in terms of letting him live at his house letting Bernard Katz and his wife live at A.V. Hill's house for about 2 years before they could get a house of their own and so on. So A.V. Hill was a very important figure in Bernard Katz's life. And who was A.V. Hill? A.V. Hill was Katz's PhD mentor at the University of London. So this is Bernard Katz, and this is Martin Gildemeister, who I said was one of his professors at Leipzig, who gave him a letter of recommendation so that he could basically join A.V. Hill's laboratory at London. And he got a Nansen pass basically, so that he could go to London and carry out his work with A.V. Hill because he was stateless at that time. And Nansen Pass basically was issued. So Nansen was actually a neuroanatomist and who had basically contributed a lot towards neuroscience. And Bernard Katz is basically a neurophysiologist. Yeah. So when Katz worked with A.V. Hill, completed his PhD with A.V. Hill at the University of London. He had come to London in around 1935, and he then did his PhD with A.V. Hill. And after that, so when during his stay in London, he basically came into he became very good friends with Eccles. So we spoke about Jack Eccles, right? So Eccles was Charles Sherrington's last student, who also made very important contributions in neuroscience. So we have spoken previously about Jack. We have spoken in various contexts about Eccles. So we also spoke in one of the lectures that Katz and Eccles had basically collaborated with each other. So so basically what happened is during Katz's stay in London for his PhD, at that time he became good friends with both Eccles as well as with uh, other scientists of that time. So because of his friendship with Eccles, Eccles invited him to join Eccles when he was working in Australia. So we had spoken about how Eccles had been working in Eccles had to return to Australia after his research assistantship at under Charles Sherrington, right? So when Eccles returned to Australia, Eccles also invited Katz to collaborate with him and do some of the work. So these are basically images of a younger version of these scientists, and then a later much older version of these scientists in Sydney. So almost 30, this image on the right is about 30 years after this initial image was taken on walking on the road in Sydney. So this was in 1942, and this is around 30 years later. So you can see that we have discussed about Stephen Kuffler in many of our previous in like sessions. Please do go back and take a look at what contributions were made by Stephen Kuffler, John Eccles, and Bernard Katz. And similarly, 30 years later, the same trio basically. So this is Kuffler, this is Katz, and this is Eccles. So yeah, so these so around for around 2 years, I think, from 1937, basically, John Eccles invited Katz to work with him in Sydney. So for around 2 years, they worked together, and after that. Katz served in the military for around 4 years during the World War, and by the time he had become a naturalized citizen of Britain. So he served in the war, and after 4 years, I think around 1943, I think, when the war got over. So at that time, he returned to the United Kingdom. So around just around when the war had finished, so Katz got married, and as a wedding present, apparently, A.V. Hill invited him back to the United Kingdom and offered him a position. So that is the story of how his the friendship of uh, Katz with Eccles. It also resulted in some of their discoveries together. So this collaboration resulted in some fruitful discoveries. Also, similarly. Katz also collaborated with Hodgkin. So during his time in London, initially when he got acquainted with Eccles, around the same time he also got acquainted with Hodgkin of the Hodgkin and Huxley fame. So he like Katz also collaborated with Hodgkin. So you can see that here he's working on the squid axon with, and together with Hodgkin. So they collaborated and they came up with the idea that sodium is involved in aspects of basically the changes the membrane potential and so on. So during that, Katz spent several summers helping Hodgkin with these experiments, and they came up with the sodium hypothesis basically. And that too because at around that time Hodgkin was getting married. So as a collegiality, basically Katz they collaborated, and Katz was helping him with his experiments on the squid giant axon. So, so this is what Katz says regarding his gratitude towards A.B. Hill, who was his mentor, that for all I have learned, not only as a pupil of a great master of experimental research, but by having served my apprenticeship with a man who never, under any circumstances, allows the deceptive counsels of human vanity to enter into your argument. With a man whose one inflexible purpose has always been the pursuit and acknowledgement of truth. So Katz was extremely grateful towards A.V. Hill because A.V. Hill was the one who offered him the opportunity to basically escape from the extremely difficult environment in Germany and work with him in London and carry out his PhD. And then after his PhD, when after his PhD was over, even after that, uh, uh, A.B. Hill also recommended him to come back and offered him a position so that he could continue his work in London and to set up a laboratory of his own and so on. So he was very grateful to A.B. Hill. So after his trip to Australia, after he served in the military, and after the war was over, Bernard Katz came back to University College London, his alma mater, where A.B. Hill was basically um, a faculty. So the war was over, but it was a very difficult time because the city was basically damaged. There was a short supply of everything, of coal, of resources, and so it was quite a difficult time for Katz to set up his laboratory. And so he often at that time it was quite difficult, but he started doing some important experiments. So one was that he started working with Hodgkin on the squid axon. The other was that there was an American called Paul Fact. So he had joined Katz, and the two of them started carrying out recordings in the at the frog neuromuscular junction. So they wanted to see, they were trying to carry out recordings at this frog neuromuscular junction and trying to see what happens. And interestingly, they observed that even in the absence of any kind of stimulation, there was still some small potentials which were being detected at the neuromuscular junction. So there was still a small amount of baseline uh, uh, potential that they were able to observe, some small amount of firing, that spontaneous potentials that they were able to observe. So they made this observation, and interestingly, they much later Katz joked about it, saying that when they saw these kind of potentials, they went back to the corridor to check whether the what they were recording was noise because of the footsteps of A.V. Hill down the corridor. So anyway, yeah, so they found Paul Fact and Bernard Katz. Basically, they found these um, spontaneous, very small amplitude, around, I think around zero point 0.5 millivolts or so, very small potentials they were able to record at the neuromuscular junction, which were spontaneous in nature. They were not, even without any stimulation of the nerve, they were still able to record the end plate potential at the muscle, these small spontaneous ones. Then Katz went on to work further on trying to understand, carried out even more detailed recordings with Del Castillo, and established, and so they called these as the miniature end plate potentials, and they established the nature of these miniature end plate potentials. They saw what happens when the nerve gets stimulated. How is that how is that potential different from the spontaneous miniature inflate potentials, and how is the so how does that so if different properties of the system are changed, how does that affect the properties of the potentials that they were observing, and so on. I'll come to that in a while. Let me tell you a little about the awards and honors that Katz received. So he was knighted in 1969. So So basically, when you say knighted, it basically means that he was conferred a title by the United Kingdom, basically. So he was conferred a knighthood. So so if you are conferred a knighthood, you are referred to as Sir. So basically he is Sir Bernard Katz because he was bestowed with a knighthood by the uh, by his country. So he became naturalized citizen of Great Britain. So he was bestowed with a knighthood for his groundbreaking work. And he also received the Copley Medal by the Royal Society for his achievements. He received the Bally Medal by the Royal College of Physicians for his excellence in physiology. And of course, since we are talking about the whole series currently is about the Nobel laureates, right? So he received the Nobel Prize in Physiology or Medicine in 1970. So he shared this with two other individuals, with whom were like those two individuals who were also working on trying to understand neurotransmission and all, but they were working independently. So these individuals were working independently, but their contribution was, you can say, more or less on the same theme basically. So they were jointly awarded the Nobel Prize in Physiology or Medicine in the year 1970. So basically, Katz received one third of the share of the Nobel Prize. So let me try to explain with a little bit of history about what Katz did and what was the significance of his work. So, beginning with Luigi Galvani, who showed that there is electricity or bioelectricity in the nerves of frogs, which can basically result in muscle contraction. So, that began the field of electrophysiology. Then uh, Otto Levy, he showed that synaptic transmission involved chemical messengers. So Henry Dale and Otto Levy received a Nobel Prize had received a Nobel Prize prior to the work conducted by Katz. So these two individuals had demonstrated that basically there are chemical messengers which are involved in neurotransmission at the synapses. So Otto Levy had shown that there is a chemical which he had called as Vegas stoph, or basically a chemical. So he had made use of the Vegas. So because of that, it was called he called it Vegas stoph. So he performed experiments with the frog heart, and he showed that Vegas stoph or acetylcholine basically slows the heart rate. And like I said, both of these individuals had received the Nobel Prize. So Henry Dale had also shown that acetylcholine is a neurotransmitter and showed that it plays a role in synaptic transmission in the autonomic nervous system. So, based on these previous evidences, basically, Katz further increased our understanding of what exactly happens at the synapse. So, how are these neurotransmitters getting released and how are they interacting with the postsynapse? So, he basically provided a mathematical as well as experimental framework for neurotransmitter release. And he demonstrated that these neurotransmitters are released not continuously, rather, they are released in discrete or separate individual packets or quanta. So, again, we have discussed about the heated debates between Eccles and uh, Dale, right? So we had discussed about how, if you remember one of the earlier video lectures, so we had discussed about how when Katz had first arrived in London, he was quite taken aback by the extremely heated debates that he had observed between H. Dale and Eccles. So H. Dale was a much more senior person, and Eccles was a relatively younger individual. But they used to have extremely serious debates regarding whether the neurotransmitters that I mean whether neurotransmission that happens at synapses is chemical in nature, that is soup, or whether it is electrical in nature, that is spark. So they used to have, so we remember, I mean, if you remember, Cards had described how extremely shocked he was at the way these individuals used to argue at these society at different conferences and meetings and so on. But beyond that, they were, I mean, as I mean, having argued about these things, they used to just go back to being normal individuals after the uh, debates themselves. So soup. The soup category was supported by Dale and Levi, and the Spark side was basically Eccles and some others. So when I say soup, I mean that these individuals believed that at the synapses, the neurotransmission happens via the release of chemicals or neurotransmitters, basically. Whereas in case of Eccles, he believed that there are probably just electrical signals which are directly crossing synapses, and there are no chemicals that are present, is what Eccles believed. But now we know that it is indeed the soup part which is true. So basically, there are neurotransmitters which are present in the presynapse, and these neurotransmitters are released into the synaptic cleft. So here, this gap is what we call as the synaptic cleft, and this is the presynaptic site, and this is the postsynaptic site. So then these neurotransmitters are basically released into the synaptic cleft by a process called exocytosis. So via exocytosis, these neurotransmitters are released. They go and interact with their receptors which are present on the membrane of the postsynaptic neuron. And then this interaction of the neurotransmitters with their specific receptors thereafter results in a cascade that causes the postsynaptic neuron to also get excited. So we now understand how this works, but at that time there was a huge debate regarding whether this neurotransmission is chemical in nature. That is basically that there are neurotransmitters such as acetylcholine and noradrenaline and basically GABA and serotonin and dopamine and so on. So now we know that these actually exist and these actually function this way. But at that time, it was not clear, it was not known. And there were individuals who were saying that what happens at the synapse is also electrical in nature, similar to what happens within the neuron itself. So, how did the soup versus sparks debate get resolved? So, basically, in different ways. So, Levi's work confirmed that there is basically, for example, he showed with acetylcholine that there are chemicals which are getting released at the synapses and which are responsible for neurotransmission. Uh, Dale also showed that acetylcholine is a neurotransmitter and it plays a role in chemical synaptic transmission in the autonomic nervous system. And finally, Bernard Katz also provided quantitative evidence of neurotransmitter release in finally ending the debate. So although Katz and Eccles had worked together and collaborated like for around 2 years, but Katz did not blindly simply like follow Eccles' side. He basically performed his own experiments, and he basically showed that neurotransmitter release indeed takes place in terms of quanta. So basically, there are neurotransmitters that are present in the presynaptic site, which are released in discrete packets, and they interact with the postsynaptic membrane, postsynaptic site, basically, resulting in neurotransmission. So basically, he provided a quantitative evidence of neurotransmitter release as discrete quanta, thereby ending the soup versus Sparks debate in favor of the soup fraction, basically. So the significance of this work was that it established the foundation for chemical synaptic transmission research. So the field, because at that time was divided between chemical and electrical synapses, so finally there was a basically it Katz's work basically kind of put the final nail in the coffin for the soup versus Sparks debate in a way. So what else did he do? So, like I said, he came back to University College London because A.B. Hill apparently gave like arranged for it to happen so that he could come back after his like he and his wife would come back after their wedding and continue to work in University College London. So he served as a professor of biophysics at UCL for a very long period of time, and during his tenure over there, he trained several generations of scientists who continued his legacy in neuroscience, biophysics, and medicine. And because of his pioneering work, his department became a global center for synaptic physiology research. So it can be regarded that the study of neuromuscular junction or the history of study of neuromuscular junction would basically be incomplete if you do not uh, mention Bernard Katz. So the way Hodgkin and Huxley are for the action potential, Bernard Katz is for the neuromuscular junction. So at the University College London, so there is basically in his memory there is a Bernard Katz building that was constructed. So in 1970, he had received the Nobel Prize along with two other individuals, Euler and Axelrod, for their discoveries concerning the humoral transmitters in the nerve terminals and the mechanism for their storage, release, and inactivation. So Katz had worked on acetylcholine. Euler, I think, had worked on noradrenaline, if I'm not wrong. So these two others had probably worked on noradrenaline. But they had basically shown that there are neurotransmitters and how these neurotransmitters are basically being released by the presynaptic membrane and how do they act on the postsynapse. So, keeping this in mind, let us go to the first question. What did Bernard Katz's research primarily focus on? So, like we just discussed, his work focused on neurotransmitter release in quanta. So, by quanta, you mean, so quantum basically means a discrete individual unit. So, these neurotransmitters he basically showed so Katz basically showed that these neurotransmitters are basically released in discrete packets or packages, and now we understand how that happens because now we understand that there are synaptic vesicles, and these synaptic vesicles can fuse with the presynaptic membrane and release their neurotransmitter content. So each fusion of each vesicle causes the release of a certain number of neurotransmitters, right? So each of these vesicle represents a quantum or a single packet of neurotransmitter release. So that is what the focus of Card's research was primarily on. Let me go back to the text again. Yeah, so we were talking about quanta, right? And what do we mean by that? So he hypothesized because I said, right. So Paul Fact and Bernard Katz had initially started carrying out these recordings, I think around 1948, 1949, around that time. So they were carried out these recordings in the frog neuromuscular junction, basically. So while doing those recordings, they saw that there are these small, very small amplitude potentials that they were being able to detect. So these, without any kind of stimulation, they were able to see these spontaneous potentials when they were carrying out their recordings from the postsynapse. So they term these as miniature end plate potentials. So miniature because they were very small, so extremely small end plate potentials they were able to observe, which were spontaneous in nature. Basically, they were happening even in the absence of stimulation. And they also saw that these MEPPs were identical in shape and timing to the evoked end plate potentials. So, what do we mean by evoked end plate potentials? Evoked end plate potentials are basically those that you can record when there has been a stimulation of the nerve. So, if there is a stimulation of this nerve, basically there's an action potential across the along this nerve, the presynaptic nerve. So that will cause the release of these I mean, now we understand that's how it works, right? So now we know that because of this action potential, these neurotransmitters will basically fuse with these synaptic vesicles containing the neurotransmitters will basically fuse with the presynaptic membrane and release these neurotransmitters. So when these neurotransmitters are released, they will interact with their receptors on the postsynaptic membrane. And because of the action potential, so because of the stimulation of the nerve, several of these vesicles shall fuse, and therefore there will be a strong potential or a much higher amplitude potential that will be recorded at the postsynapse. So these uh, end plate potentials that are recorded when the presynapse has been stimulated or the nerve has been stimulated. In that case, the end plate potentials that are recorded in the muscles are called as the evoked end plate potentials. So they observed that, so like I said, Paul Fatt and Katz had started this work, and Katz continued this further with Del Castillo. So they carried out extremely detailed analysis of these recordings and so on, and they saw that these miniature end plate potentials, the spontaneous ones, were identical in both shape as well as timing to the evoked potentials, basically showing that these MEPPs were responsible for the when there are MEPPs, basically there is a release of there is basically quantal release, or basically there are probably one that each time as a single synaptic vesicle is randomly fusing with the membrane, for example, we are able to detect that as the MEPP at the postsynapse or the muscle. So because of they were able to see these kind of MEPPs, these spontaneous MEPPs, and because these MEPPs were identical in shape and timing to EPPs, so they came up with this idea, or they came up with this hypothesis that neurotransmitters are released in discrete packets or quanta rather than as a continuous flow. And the choice of neuromuscular junction that they, I mean, the reason they worked on neuromuscular junction because at that time, the central synapses was a complete black box. It was very difficult for people to study the central synapses. It was extremely challenging. But the neuromuscular junction were much larger in size, and it was much easier for them to actually record from the neuromuscular junction and observe the neuromuscular junction. So, this large size as well as the simplicity of the neuromuscular junctions made these structures ideal for studying these kind of synaptic events rather than the central synapses. So, what was the actual experiment they performed? So, the actual experimental setup was as follows. So, the frog and rat neuromuscular junctions were dissected, and microelectrodes or extremely small electrodes were used to measure or detect the small changes or fluctuations in membrane potential. So what did they do? They are first isolated the muscle fibers, then they stimulated the presynaptic nerve. So, for example, this is the presynaptic neuron, right? So they stimulated this presynaptic nerve. Then they observed that, then they recorded both the evoked potentials, so the evoked end plate potentials as well as the spontaneous miniature end plate potentials. So they recorded both of them, right? So here you can see the top one is the evoked end plate potential, okay, and the bottom is the miniature end plate potential. So you can see that these are extremely, if you compare the scale, you will see that this potential difference, right? This potential, like compare this potential difference versus this potential difference. So extremely small, around zero point 0.5, you can say, around 0.5 millivolt of a difference versus around 50-ish, 50 to 60 of a difference, right? So you can see that the evoked inflet potentials are, because they are caused by the fusion of several of these neurotransmitters. So these are basically because of a lot of these synaptic vesicles fusing with the presynaptic membrane. So that is the reason why the MEPPs and EPPs were identical in shape and timing, because the EPPs were basically the summed potentials of the MEPPs. So they observed that, and they also varied calcium concentration to study its effects. So that came later. So the changing the calcium concentration, that the experiments with calcium basically came a little later. These were the initial experiments that they were doing. So, what did they observe? They observed that these EPPs are basically integer multiples of MEPPs. So how does that make sense, or how does that explain the quantal hypothesis that they were putting forward? So, if so, the MEPP, let's assume, say, the MEPP has so there are n number of neurotransmitters that are getting released by the, just a minute, yeah. So there are n number of neurotransmitters that are present in that are getting released by at once. So there is that are present in one synaptic vesicle, right? So whenever there is a fusion of the synaptic vesicle with the presynaptic membrane, it is not possible for only half a synaptic vesicle to fuse, right? So only few of the or only few of the neurotransmitters to get released, right? Because the synaptic vesicle is fusing with the presynaptic membrane. So all of the content of that of that synaptic vesicle is going to get released. So each MPP will correspond to the release of a synaptic. Let's assume that each MPP corresponds to the release of a synaptic vesicle. So now when during an evoked end plate potential, what happens is several of these synaptic vesicles are basically fusing. So let's assume there are, say, m number of synaptic vesicles that are fusing. So now the final EPP that they are going to get is basically going to be M into N, correct? Because initially for one vesicle fusion, if the potential that you're getting, or the result that you're getting is N, then if there are M number of vesicles that are getting released, so the final value that you'll get will be M into N. So that is what they observed, that if one vesicle is getting released and that one vesicle has n number of molecules, and during a stimulation there are v vesicles that are fusing with the membrane, so the total number of molecules that are going to get released is going to be v into n, so v times n. So if one vesicle is getting released, then it's going to be just n. If there are v number of vesicles, it's going to be v into n. Therefore, the the potentials that we are going to, so these are the neurotransmitters, right? And these neurotransmitters are going to bind to the postsynaptic, there are specific receptors in the postsynapse. So the potentials that they are going to, the potentials that they are going to evoke are also going to be integer multiples of the uh, potentials that are observed when one vesicle is fusing with the presynapse. So the EPPs will be integer multiples of the MEPPs. So that's what they observed, that EPPs are integer multiples of MEPPs, supporting the quantal hypothesis. So later, Katz also performed some experiments with his colleague, and the two of them showed that when there is a change in the calcium levels, then also the one of the effectors of synaptic vesicle fusion is basically calcium. So they varied the calcium concentrations and they studied the effect of that on the MEPPs and EPPs. And they saw that if the calcium is decreased, then the number of neurotransmitter quanta that is released decreases. So calcium is required for the fusion of synaptic vesicles with the presynapse. So if calcium is present, then it will enable the synaptic vesicle to fuse with the presynapse and the neurotransmitters to get released. So that's why reducing the calcium decreased the number of neurotransmitter quanta released. What else did Katz do? So, so like we discussed before Katz's research, there was a lot of debate about how nerve cells and muscles communicate with each other. There were people like Eccles who believed that there was direct electrical transmission, whereas those like Henry Dale and Leovi, they showed that, they believed and they put forth evidence to show that it was a chemical process. And Katz basically confirmed that it is indeed a chemical process, and he used intracellular recording techniques. So he recorded at the neuromuscular junction. He recorded at the postsynaptic muscle basically, and he showed that the a neurotransmitter that is responsible for synaptic signaling at the neuromuscular junction is basically acetylcholine. And he also showed that the release of this acetylcholine happens in a quantal manner, or that certain discrete packets of acetylcholine get released uh, for the neurotransmission to happen. And how was his work helpful? How did it help neuroscience in general? So his work basically, first of all, helped settle the debate between the electrical and chemical transmission theories, and supported Sir Henry Dale's hypothesis of chemical neurotransmission. And it also helped in paving a way for modern synaptic physiology. So even now, when we carry out electrophysiological recordings at the neuromuscular junction, we record the MEPPs and the EPPs, and we calculate the quantal content, and we determine whether certain mutations or certain changes or certain kinds of drugs can alter these phenomena. So that this in his discovery of MAPPs and his discovery of the quantum nature of neurotransmitter release was extremely important and continues to influence the field of the area of electrophysiology basically. So, like I said, Katz and his colleague Fat carried out the MEPP recording in the NMG of skeletal muscles, and they observed that there were these small spontaneous electrical signals which occurred even in the absence of nerve stimulation. And because of that, they were able to hypothesize that neurotransmitters are not released continuously but in fixed, discrete quanta or packets. And it also helped us to understand why neurotransmitter release is extremely reliable and precisely regulated. So, when there is a signal that these neurotransmitters have to be released, only then the synaptic vesicles fuse with the presynapse. And that also involves a lot of elaborate machinery. It involves the snare complex proteins, and a lot of machinery is involved for the correct sequence of events that are required for neurotransmitter release. It also provided the first experimental evidence that nerve signals are transmitted in fixed units. And of course, currently I have been explaining everything in terms of synaptic vesicles for your easy understanding. However, at that time, when Katz and Fat were carrying out these recordings, all they were able to observe were these signals. Right, so that we discussed about how they were able to. So this is an original image. This is an image from Card's Nobel lecture. I highly encourage you to, I strongly encourage you to go back and read his Nobel Prize lecture. It is written in a very, easy to read manner, and remains relevant even today. So on top you can see how they how they were able to record the miniature end plate potentials basically, and here the bottom refers to the evoked end plate potentials. So at that time they were only able to make these recordings, and around the same time, around in the 1950s, that is when we know that Pallad and Pele and others had were starting to use basically since the electron microscope was the advent of electron microscope happened in the around 1950s, and people were trying to look at different kinds of biological preparations. So, and we had discussed about how Pallad and Pele were looking at the synaptic, the basically they were the, Pele observed the synaptic vesicles, and they showed, this happened around the same time. So these recordings by Katz and were basically, so they understood that there might be a link between these recordings and the synaptic vesicle structures they were able to see. So this using these recordings and correlating that with the ultrastructural information. So Katz inferred that these were probably because of the nature of the neurotransmitters being packed in the synaptic vesicles for fusion with the presynapse. So it was later confirmed that these quanta corresponded to the contents of synaptic vesicles, linking the electrophysiological findings of Katz and Fat and Del Castillo basically to the cellular structures or the ultrastructural images that were observed by Pallard and Paley and others. So when they were actually doing the recordings, of course, they did not know about the presence of these kind of synaptic vesicles. I mean, they after carrying out these recordings, and during the same time when other researchers were showing that these vesicles might exist, so they were able to correlate these electrophysiological findings to the presence of those structures. So next, cards. Having understood that there are these synaptic vesicles, because of the work of others, Katz proposed that these synaptic vesicles probably store the neurotransmitters and release them into the synaptic cleft during neural communication, which we now know to be actually true. So his research demonstrated that the neurotransmitter release correlates with vesicle fusion at the presynapse, and electron microscopy later confirmed that vesicles exist and transport neurotransmitters. And he also showed that the number of vesicles released determines the strength of the synaptic signal. So basically, if more vesicles are released, the signal is stronger. I mean, of course, we understand that when more vesicles are released, more neurotransmitter is released, and more of it goes and interacts with the receptors, and that results in the downstream signal in the postsynapse. And of course, it helped us in better understanding how different kinds of drugs and toxins can affect neurotransmitter release. And of course, because we understand that these kind of neurotransmitters are released into the cleft, and that is how synaptic transmission happens. In many cases, for example, in case of, say, treating depression with selective serotonin reuptake inhibitors, because we know that these neurotransmitters are getting released, and we know that there are these reuptake mechanisms that exist for taking the neurotransmitter back into the presynapse, we are also therefore able to come up with these kind of for example, SSRIs for treating diseases such as disorders or problems such as depression. So, of course, Card's work was extremely seminal in nature. Then, of course, like I said, Del Castillo and Cards tried to understand. So, having understood that these neurotransmitters are getting released into the synaptic cleft, they next try to understand how these are actually activating the postsynapse. So, what did they see? So, one is the Del Castillo and Katz model of receptor activation. So they developed a two-step model for the way neurotransmitters actually activate their receptors. Now this was extremely important because before Del Castillo and Katz model, previously it was assumed that neurotransmitter binds, receptor activates immediately. But their experiments showed that neurotransmitter binding is actually only the first step. They proposed that there must be a conformational change of the receptor upon binding of the neurotransmitter. So basically, the neurotransmitter binds to the receptor, then the receptor undergoes a conformational change, which is the second step, and this conformational change of the receptor basically triggers the activation of the postsynaptic neuron. So this model helped scientists to better understand how receptors actually function, and of course influence the development of drugs for blocking the neuromuscular junction. For example, we know that in myasthenia gravis, for example, so that is, I mean, it's a very, I will leave that to you. Go back and check myasthenia gravis and what kind of treatments are being explored for that disease, basically. Next, his work laid the foundation for modern receptor pharmacology, including research on ligand-gated ion channels. Yeah. Also, like I said, that Katz worked with one of his colleagues at University College London, Ricardo Malady, and the two of them basically showed that calcium ions were a key trigger for neurotransmitter release. So they showed that if calcium is not present, so let me go back again to. Yeah. So again, this is taken from Katz's Nobel Prize lecture, and here you can see. So, if so, they used what they called a calcium micropipette. So basically, they immersed the frog sartorius muscle in calcium-free solution containing 0.84 millimolar magnesium, and they filled a micropipette with zero point 0.5 molar calcium chloride to record the focal external potentials from a localized junctional spot. And they were controlling the efflux of calcium electrophoretically. And they performed, and you can see four different recordings. And so in the first case, there was no calcium. So the calcium efflux was blocked. So here there is no calcium, and you can see that uh, there is no neurotransmitter release. In case of B, where there is a little calcium, so in that case there is, you can see that there is a little I mean there are certain there is certain release of neurotransmitter. So basically they are saying that single transmitter quanta were released in some cases. So in around three trials, they were able to see, so one, two, and three, in three different trials, they were able to see single quanta release of neurotransmitters when there is a little calcium present. Third, when the calcium is increased, so there's a little more calcium, they were able to see even more increase in the quanta of neurotransmitter release. Then fourth, again, when the calcium was blocked or not present, again it stopped. So neurotransmitter release stopped. So they showed that calcium is necessary for the release of neurotransmitters. So using a focal calcium injection technique, the one that we just discussed, they showed that when calcium enters the presynaptic neuron, it basically triggers the vesicle fusion and thereby the release of neurotransmitters. And when calcium was removed from their extracellular environment, the nerve impulses could no longer trigger neurotransmitter release. Basically, what we just discussed. So this was done a little later by Katz together with his colleague Malady, and they showed that again a very important discovery that calcium influx is necessary for synaptic transmission. And it also explained why calcium channel blockers can modulate nerve signals. And this is crucial for treating neurological disorders and pain. So we know that people who have neuropathic pain or people who are having chronic pain issues, in those individuals, like certain therapeutics or drugs can be developed to modulate nerve signals on the basis of calcium channel blockage. Moreover, it also helped in treatment of diseases such as epilepsy, Alzheimer's, and Parkinson's. Then, in later years, Katz studied acetylcholine noise, and he showed that there were tiny fluctuations in electrical activity caused by the action of neurotransmitters on the receptors, and these were due to random opening and closing of individual ion channels. And by analyzing these extremely tiny fluctuations in electrical activity caused by the uh, action of neurotransmitters on their receptors, they deduced important properties of the receptors for these neurotransmitters. And this work basically set the stage for the patch clamp technique, which was developed by Nehrer and Sakman. So Sakman was again a mentee of Bernard Katz. So Sakman was a mentee, he was a student or a mentee of Katz, who later went on to develop this patch clamp electrophysiological technique, and again won the Nobel Prize for that, because they were able to use this to measure even to the resolution of single ion channel currents. Like I said, he had collaborated with Hodgkin in around 1948, around at that time, and investigated the role of sodium ions in nerve impulses. And this basically led to the sodium hypothesis, which was a precursor to the Hodgkin-Huxley model. So these are all the contributions of Bernard Katz, and this is the paper in which they have described the way they perform the experiments in the frog neuromuscular junction. And I'll not go into this in, in too much. I'll not go into this in too much detail because we have discussed most of these. But if there is time at the end of the class, we might come back to this. So now going back to the questions. So, second question, which neurotransmitter did Card's research focus on at the neuromuscular junction? So, we discussed that Card's work showed that it is acetylcholine, which is present, which is a neurotransmitter at the neuromuscular junction in mammals at least, and I mean. Also I think in I think amphibians and so on. So he showed that it is the neurotransmitter acetylcholine, which is released at the neuromuscular junction. So it is first packaged into the synaptic vesicles. These vesicles fuse by exocytosis. Acetylcholine binds to its acetylcholine receptors on the postsynapse. As you can see, the postsynapse is very like highly folded structure basically to increase the surface area. So, when it is released, it binds to the postsynaptic acetylcholine receptors, and that triggers a conformational change and subsequent activation of the postsynapse. So the end plate potentials that they recorded basically. So this is the structure of acetylcholine. I hope that is clear to you. Next question. What was the key experimental observation supporting Card's quantal release hypothesis? So we discussed that when Cords and Fat were making these recordings, they saw that in the absence of any stimulation of the presynaptic nerve, they were still able to record these miniature end plate potentials in the postsynaptic muscle. So, even in the absence of any stimulation, there were these spontaneous release of the neurotransmitters, and these were more or less fixed in amplitude. So you can see that the peaks are more or less of similar height, right? So these were more or less fixed amplitude and spontaneous. They were random. They did not originate from any particular region or something. So these kind of potentials, when they observe, they were able to understand that when there is actually stimulation of the presynaptic nerve, and they when they happen to record these evoked in plate potentials, those are actually because of the increased frequency and rate of fusion of, I mean, increased rate and frequency of the release of the neurotransmitters. So initially the I mean, in the absence of any stimulation, these neurotransmitters are released in small discrete quanta, because of which in a spontaneous random manner, and because of that we are able to see these miniature end plate potentials. And in case of stimulation of the nerve, there is an increase in the frequency and rate of release of the neurotransmitters, again of the same discrete packets or puncta of, quanta of neurotransmitters, but there is an increase in the frequency and rate of release of them. Because of that, we are able to record these summed potentials of which they called as the evoked end-plate potentials. And these EVP, these evoked end-plate potentials were integer multiples of these MEPPs, which basically showed that the neurotransmission is quantal in nature. So the key experimental observation supporting Katz's quantal release hypothesis was the observation of these spontaneous end-plate potentials. Next question. What role did calcium ions play in neurotransmitter release according to Katz? So, like we discussed, when calcium is absent, neurotransmitter release does not happen. When calcium is present, neurotransmitter release happens. And when calcium I mean, calcium concentration increases, there is even higher or even excuse me. So when calcium concentration increases, there is an increase in the release of neurotransmitter release. And again, when calcium is removed, again the neurotransmitter release also stops. So what role did calcium ions play in neurotransmitter release according to Katz? The calcium ions basically triggered the neurotransmitter vesicle fusion with the presynaptic membrane, resulting in exocytosis of the neurotransmitters. So I hope so far it is clear to you. If you have any questions, feel free to put them in the chat box or unmute yourself and ask me. Okay, I gather that no one has any questions. Moving on to the next uh, previous year assignment question. Which experiment provided evidence for the quantum release hypothesis? So we just discussed this, that recording of the miniature end plate potentials in the neuromuscular junction was responsible for providing the evidence for the quantal release hypothesis. So the way Del Castillo and Katz have described this in their 1954 paper is as follows. So what they say is that suppose we have at each nerve muscle junction a population of n units capable of responding to a nerve impulse. Suppose further that the average probability of responding is P. So remember, there is always a, so when there is a resting state also, when the nerve is not stimulated also, these MEPPs are getting, no, these MEPPs exist, right? So there is always a release that is happening. And when there is a stimulation of the nerve, the probability of the neurotransmitter release increases, right? So the neurotransmitter release is happening at all times, even when there is no nerve stimulation. Even then the neurotransmitter release is happening. But when there is stimulation, the probability of release increases, basically. The probability of physical fusion increases. So now, coming back to this, so suppose further that the average probability of responding is P. Okay, so then the mean number of units responding to one impulse is M is equal to NP, right? So N is the population of units, the population of units which are capable of responding to a nerve impulse, right? And the probability of responding is P. So the mean number of units that are responding is going to be N into P. So when P is very small, so I think I'm quite sure many of you would have heard of Poisson's law and Poisson's distribution. If you have not, please go back and check this. So basically in case where the probability of an event is very small, so the distribution is described, and the distribution that we take into consideration for these kind of events is the Poisson distribution. So if in case you are interested, please go back and check the Poisson's law and the Poisson's distribution. So based on that, what they are saying is that when P is very small, the number of units X which make up the EPP in a large series of observations should be distributed in the characteristic manner of Poisson's law. So even if this goes over your head, please do not worry. It is only I just wanted to give you an understanding of how Del Castillo and Cards basically approached the problem. So they only had these recordings. They did not have any other idea. They did not know about the synaptic vesicles when they were doing this, the existence of these synaptic vesicles when they were carrying out these recordings, and all they could do was come up with a mathematical model or a mathematical understanding. So as you can see, they are trying to establish a statistical understanding of how these neurotransmitters were getting released. So I just the idea of showing you this passage was to only explain to you that by using their recordings and their understanding of statistics and mathematics and their intuitive thinking, their logical mindset, basically, they came up with how these neurotransmitters are getting released and showed that it happens in a quantal manner. That is all. So please do not get confused with this passage. It is not, it is not important for your course as such, but I was just trying to explain how Katz approached this problem and gave us a quantitative understanding, a quantitative model of how neurotransmitter release happens at the muscular junction. So coming back to the question, which experiment provided evidence for the quantum release hypothesis? The recording of miniature end plate potentials at the neuromuscular junctions. Next question. Okay, I'll give you a little background about Roger Sperry first, and then we will discuss about these questions which deal with Roger Sperry's Nobel Prize winning work. So now, Roger Sperry was an American neuropsychologist and neurobiologist. So interestingly, Sperry initially started, so uh, he was born in Connecticut. He first started with a degree in English. So he started with a degree in English, and while studying for his degree in English, he came into contact with certain researchers and certain faculty and so on who were working on psychology. So that actually increased his fascination and his he started wanting to learn psychology instead. So he, after doing a bachelor's in English, he actually went on to do a master's in psychology. And while doing a master's in psychology, he realized that he was interested in understanding pursuing this further. And then he carried out a PhD in zoology from the University of Chicago. So, under Paul Weiss. So, we have discussed about Paul Weiss previously. When we were discussing about the signaling molecules in one of the very early lecture videos, when the signaling molecules like NGF and so on were discussed, at that time we had spoken about Paul Weiss. So Paul Weiss had given the contact guidance theory, if you remember. And his PhD student was Roger Sperry. So Roger Sperry carried out his PhD under Paul Weiss, who was a neuroembryologist basically. So he did a PhD in zoology from the University of Chicago under Paul Weiss, and then he went on to do a postdoctoral studies with Carl Lashley at Harvard University. Interestingly, uh, Roger Sperry when we discussed about Roger Sperry and Carl Weiss and Lashley, when we're talking about the signaling molecules, we had discussed about how Paul Weiss had both Paul Weiss and Carl Lashley were both individuals who thought that, whose idea was that the connections within the brain were non-specific. They were not specific in any way. It was all just a bunch of non-specific connections is what they had proposed. But Roger Sperry went on to, sorry. So Paul Weiss had come up with what is called the contact guidance theory. So by just mechanical contact or mechanical interactions, they neurons establish connections with their target cells is what Paul Weiss had proposed. But Roger Sperry, who who had been a student of Paul Weiss and who had been a PhD student of Carl Weiss and had done his postdoc with Carl Lashley. He came up with the chemi affinity hypothesis. I'll come to that in a while. So, okay, no, let me explain that right. Yeah. So he came up with the chemo affinity hypothesis, basically, which was in complete contradiction, which completely was the opposite of what his PhD supervisor and his postdoctoral supervisor had proposed. So they had both proposed that these connections are non-specific. There are no specific connections that exist in the nervous system between these neuronal cells. They probably have these kind of mechanical interactions and so on. That's it. But Roger Sperry proposed, you can see in this 1963 paper in PNAS, in which he is the sole author. So he had proposed that there is a precise and ordered growth of nerve fibers and their synaptic connections. So basically, nerve fibers probably have certain specific chemical signals or bioidentification tags, or what he called as chemoaffinity. And these are not just mechanical or random processes as proposed by others, including Weiss, and that these these chemical identifiers or these bioidentification tags are basically what helps the neurons and their axons to establish extremely specific connections with their target cells. So Sperry carried out a lot of experiments in salamanders and newts, and basically in organisms in which the regeneration capacity is very high. So basically, he would cut the nerve fibers, he would rotate the eye by 180 degree. So by cutting the optic nerve and rotating the eye by 180 degree, then and then after the regeneration of the nerve had happened, he would observe which way, like whether the connections were being formed uh, in the same, like with the same target, or whether the connections were uh, being formed in a random way. And he observed that the connections were extremely specific. So even after the eye had been rotated 180 degree, still the uh, nerves had formed connections with their exact same cognate targets. And therefore the animal was actually, because the eye had been rotated 180 degree and then regeneration had happened, the view of the world for the animal was actually upside down. So he showed by these kind of experiments, and these and several other experiments basically, that the synaptic connections and the way the neurons home in on their targets is extremely specific, and this is probably due to the presence of certain chemicals. He also showed that these kind of, I mean, he also speculated that these like these kind of specific guidance that happens is probably because of a chemical gradient that exists in a spatial manner. So there's probably a spatial gradient of chemicals which directs the neurons to their targets. So depending upon the chemical gradient and the specific kind of like tags and the receptors for those tags that are present in the neurons and their targets, uh, the growth of the neuron towards its target takes place and the connections are established. So this is the chemoaffinity hypothesis that was proposed by Sperry, which, like I said, contradicted or was basically overthrew both his PhD supervisor as well as his postdoctoral supervisor's hypothesis. So Sperry argued that even if mechanical forces like contact guidance were at play, they could not explain the selectivity observed in nerve growth. So like I said, the contact guidance theory was that proposed by his PhD supervisor, Paul Weiss. And he posited instead, so Sperry posited that chemical gradients, such basically morphogenetic fields, probably influence the development and organization of neural connections far beyond what mechanical explanations could account for. And now we do understand that this does happen in the nervous system. So when the axonal growth cone is traveling towards its target, it is guided by certain chemical gradients, by certain cues, and by the presence of certain tags on its target, and so on. So this indeed was extremely important. And this is true, for example, in the retina, in the vestibular system, and so on. and it also helped in explaining the topographic mapping of neural systems. So there is a map of the how the retinal fibers map exactly to corresponding regions in the brain because of the specific interactions and the formations of extremely specific connections. So of course, when Sperry proposed, all of this was just a hypothesis, but later in the 1950s and 1960s, actual histological studies began to validate his hypothesis. Like I said, Sperry and his colleagues carried out a lot of experiments on different animals, especially those which were capable of regenerating their organs, such as salamanders and newts and so on. And they showed that there are that for example, in the optic system, the regenerating fibers follow very precisely mapped chemical cues to ensure the correct topographic arrangement in the brain. And of course, I explained to you about the axon guidance molecules, so how they actually behave in the way Sperry had predicted the chemoaffinity hypothesis to work. So that is all about the chemoaffinity hypothesis of Sperry. But this is not what won him the Nobel Prize actually. Uh, what actually won him the Nobel Prize was his work on the split brain. So what he had done was that he had taken, I mean, he had used both animals as well as human patients to understand whether there is a specialization between in terms of functions between the two different hemispheres of the brain. And what had he observed? So in patients, during those days, in patients who are having very severe forms of epilepsy, which were not being managed in any way whatsoever, the in some patients, not in all, in certain patients, the corpus callosum was cut or severed. So the surgeon used to sever the corpus callosum. So what is the corpus callosum basically? So we're talking about the two different hemispheres of the brain, right? So the corpus callosum basically kind of uh, was kind of served as a bridge between the two different hemispheres. So basically, it allowed the transfer of information between the two different hemispheres. So, those patients where the corpus callosum was severed for the management of epilepsy, Sperry and his students showed. So Sperry and one of his students Myers. So they showed that when the corpus callosum was severed, patients could not process, so they were not able to communicate the information that was being processed by the different hemispheres. So he showed that certain functions are more so the certain functions are carried out more by one of the hemisphere, and certain other functions are carried out like more dominantly by the other hemisphere. For example, he saw that the left hemisphere is more specialized or is more dominant for activities such as speech, reading, writing, and logical reasoning. And so, if you, like for example, uh, if the right hemisphere in those patients, if the right hemisphere was provided with some information, the because the corpus callosum was severed, the information could not pass to the left hemisphere, right? So they were the patients were not able to describe the information that they were being presented with. So they were, because the left hemisphere is responsible for speech and language and so on, but only the right hemisphere was getting the information. The individuals were able to identify the object. They were able to identify the object, or they were able to identify the stimulus, but they were not able to verbally communicate that, right? So they were able to, for example, if you showed them that object such that only the right hemisphere was activated, they were able to draw the object, or they were able to select the object with their left hand, but they were not able to describe the object verbally. Why? Because the left hemisphere is responsible for speech. And because the corpus callosum was severed, the left and the right hemispheres were not able to communicate. And whatever information was processed by the right hemisphere could not be received by the left hemisphere, and therefore the speech, or the verbally the individual could not identify the object. Similarly, he saw that the right hemisphere is involved in visual spatial processing, emotion, and facial recognition. So, uh, he saw that the right hemisphere controls non-verbal functions, so the emotional aspect, the creative aspects, the like face recognition aspects, processing this the spatial environment, and so on. So all of that was controlled by the right hemisphere. So this showed that there is hemispheric specialization that is involved. So he carried out experiments in animals such as cats and so on. He also did the same with human patients in whom the corpus callosum was severed. Then he because b- there was a disconnection between the hemispheres, he wanted to understand whether the patients felt like they were still one person, or whether they felt like there was a disconnect, or whether there were multiple individuals or something like that. So, despite the severed connection between the hemispheres, patients reported feeling that there was still one single unified person. So, Grazinga proposed the interpreter theory to explain this. So, he was Sperry's colleague who proposed that the left hemisphere, so in these individuals, probably the left hemisphere plays a role as the interpreter of actions initiated by the right hemisphere, creating a narrative to make sense of those actions. So, because there's a disconnect and the left hemisphere has no access to what the right hemisphere is doing, whatever actions that the right hemisphere is initiating, the left hemisphere does not know, is not getting the information because the corpus callosum is severed. But in these individuals, the left hemisphere is probably fabricating a story or interpreting the actions of the left hemisphere to make a story, to make a sense of why the right hemisphere has done that particular kind of action. So it's basically acting as an interpreter of the actions that were carried out by the right hemisphere, because it does not actually have direct access to that information. In these patients who have a hemispheric disconnection because of the severed corpus callosum. So, what effects they saw in these individuals who had a split brain, or basically these kind of patients? They saw that these patients often exhibited conflicting actions. So, because there was a disconnect between the different hemispheres, when one hand was reaching for an object, the other hand was resisting. Why? Because the left and right hemispheres could not communicate with each other. And So basically, the left hemisphere controls the right hand, and the right hemisphere controls the left hand. Okay, so it is inverted. The right hemisphere controls the left hemisphere controls the right hand. Similarly, the right hemisphere has access to the left visual field, whereas the left hemisphere has access to the right visual field. So if you see this image, the connections are opposite. So the left hemisphere has access to the right visual field, whereas the right hemisphere has access to the left visual field. Okay, so if you basically made the animal or the individual to close one of their eyes and presented an object or something to only the other visual field. For example, if the individual was made to, I mean, something was presented only to the left visual field, right? So the if the left visual field is getting the is being shown the object, the right hemisphere will process that information, correct? So we discussed that the left visual field is controlled by the right hemisphere. So the right hemisphere will process that information. The right hemisphere is responsible for visual and spatial processing, emotion and facial recognition, right? So you can like they were when the individuals were able to, because they had visual and spatial processing and facial recognition and emotion, they were able to draw it or they were able to select it, but they were not able to verbally identify the stimulus. Why? Because that is actually the job of the left hemisphere, and the left hemisphere is being kept in the dark. You're not showing the left hemisphere. You are not showing it to the right visual field. If the right visual field is not getting access, the left hemisphere is not able to process it. So the left hemisphere does not know. But they were able to draw or select it with their left hand. Why? Because again, the left hand is controlled by the right hemisphere. So now what is the problem with that? The problem was that in those individuals, what happened is because the two hands were being controlled by different hemispheres, the two visual fields were being connected by different hemispheres. So in those individuals where the corpus callosum was severed, when one hand was reaching for an object, the other hand was resisting. Why? Because the two hemispheres are not communicating. Similarly, when you had to button a shirt, so there was a bimanual coordination was required. Both hands are required. That was a problem. Again, why? Because the two hemispheres could not synchronize their action. And we know one hemisphere was responsible for one hand, the other hemisphere was responsible for the other hand. But it was seen that over time, many patients adapted to this separation. Although there was some confusion and difficulty in performing tasks where there was a lot of integrated hemispheric coordination required, but otherwise, many patients did adapt to the separation, which shows that there was neuroplasticity involved and there was adaptation that was being mediated by the nervous system in these patients. So, despite the severing of the corpus callosum, these patients reported feeling whole. Why? Because the theory that was proposed to explain is that the interpreter theory by Michael Zalaniga, who showed that, who proposed that the left hemisphere is probably creating a logical story or a narrative to explain the behavior of the right hemisphere so that there is an illusion of a unified self. So nowadays, of course, no one actually carries out the corpus callosum. Like no one actually carries out any surgery to sever the corpus callosum because that's a very risky surgery. So that no longer is carried out in humans at least, and techniques such as fMRI and PET have become the primary methods to study brain activity and to study the role of different hemispheres in different kinds of activities. So for his work uh, on this the split brain and the hemispheric specialization, Sperry was awarded the 1981 Nobel Prize, which he shared with David Hubel and Torsten Wiesel for discoveries related to brain function and visual processing. So his work was fundamental in understanding brain lateralization, which is the idea that different functions are located in each of the hemisphere of the brain. Also, he also worked to understand the how consciousness actually works. So let me tell you a little about that, and I'll go back to the previous year assignment questions. Yeah. So, Sperry tried to, based on all of these experiments and his other understanding, Sperry wanted to understand how consciousness works. And he proposed that the mind and the brain interaction is not dualistic in nature. So he said that it is a monoistic, uh, uh, it is not dualistic, it is monoistic. So what does that mean? So he meant to say that so like at that time the prevailing view was that mind and body are separate and mind and body are basically dual. So there are two individual separate things, mind and body. That was what was the prevailing notion at that time. So he said people what the materialist view in neuroscience at the time was that there is mind, there is body, and conscious experience was completely ignored. So the materialist view said that experience does not actually play any role on in influencing brain activity. So brain function was completely neurophysiological. There is no role of consciousness or on influencing brain activity in a way. But Sperry said that consciousness is an emergent property of brain activity, and this emergent property, so this mind, which is an emergent property of brain activity, is also playing a causal role in shaping brain process. So it is emerging out of brain activity. So the mind is emerging out of brain activity, and it is also influencing future brain activity. So brain activity is creating the mind, and this mind is based on the experience of the individual, based on consciously what that person is experiencing. This mind is actively also shaping the brain activity. That was what Sperry proposed. So historically, what existed was the idea of dualism, which stated that mind and body are separate entities, right? And this basically led to the idea that there is an immaterial mind which exists beyond the physical brain. That was what the historically the idea of dualism that had existed. Sperry proposed a monoistic uh, idea in which he said that mind and body are not separate. Rather, the mind or the mental phenomenon is actually an emergent property of brain activity. And he said that it should be understood that this mind or the mental phenomenon is inseparable from brain activity. Rather, it emerges from brain activity. And he asserted that consciousness is actually a part of the same continuum as brain function. So consciousness also arises because of brain activity, but this consciousness and the mind also influences the brain activity from which it arises. Moreover, uh, mentalism versus materialism. So, again, traditionally, materialism or behavioralism held that everything, including mental phenomena or mind, can be explained in terms of physical processes and neural correlates. And behaviorism argued that consciousness does not play any role in behavior, and mental states were all epiphenomenal or irrelevant. But Sperry's view was that, Sperry says that there is mentalism, which says that the subjective experience of the individual is not just a passive reflection of the neural processes. Rather, this subjective influence actually plays a causal active role in influencing brain activity. So it is not just a physical process, it actually plays a causal role in influencing further brain activity. So, Sperry's monoistic theory of mind brain interaction uh, counteracted dualism, and currently his theory of mind brain interaction basically aligned much more with the modern outlook of neuroscience as compared to the dualistic theory of mind brain, basically in which mind and brain were considered to be separate. So, like I said, consciousness is increasingly recognized as a legitimate topic of scientific inquiry and as a causal force in cognitive and behavioral processes. And it is integral to brain function and cannot be explained by neurophysiological activity alone. So, now going back to the previous year assignment question, what was the focus of Roger Sperry's Nobel winning research? His focus was on studying the effects of severing or cutting the corpus callosum and understanding the specialization of the two cerebral hemispheres. So the left and right hemispheres, whether they are having any specialized functions, whether they are having any, is either of them more dominant in processing certain kind of stimuli, and so on. So this is Sperry at his desk. You can see that there is a drawing of the hemispheres of the brain, lots of images of the brain. And like I said, he received the Nobel Prize in 1981 for his discoveries concerning the functional specialization of the cerebral hemispheres. So, what did Sperry discover about the left hemisphere of the brain? He discovered that the left hemisphere is primarily responsible for language and logic. So, we discussed this, that the left hemisphere is responsible for language, logic, any kind of like writing, speech, and all of that, right? Just let me go back. Yeah. So the left hemisphere is dominant for speech, reading, writing, and logical reasoning. And the right hemisphere is involved in visual spatial processing, emotion, and facial recognition. So these other options basically all refer to the right hemisphere of the brain. Whereas the language and logic are the processes that are basically processed by the left hemisphere. Next question. What concept did Sperry's split brain research lead to regarding consciousness? So, the concept of his research showed that consciousness arises from the interaction of two independent cognitive systems, right? So, it is not controlled by the left hemisphere alone, it is not controlled by the right hemisphere alone, right? It is relevant to brain function because we know that we just discussed that Sperry proposed that consciousness is an emergent property of brain activity, and it also it plays a causal role in brain activity, right? So it is not irrelevant to brain function. That is not correct. Consciousness is a unified process in the brain. So we just saw that in these individuals, just a minute. We saw that in these individuals, what happens is that because because of the severing of the corpus callosum, although they reported feeling whole as a unified self, there was a lack of communication between the left and the right hemispheres, right? So the left and right hemispheres did not actually know what the other hemisphere was actually doing, or what the other hemisphere was actually the what actions the other hemisphere was actually initiating. For example, the left hemisphere did not know that what behavior is being initiated by the right hemisphere, correct? But the left hemisphere was basically creating it was basically fabricating a story or a narrative to explain what the right hemisphere was doing to preserve the illusion of a unified self, right? So from this, what do we understand? We understand that consciousness actually arises from the interaction of two independent cognitive systems. The left and the right hemispheres are two independent cognitive systems which interact with each other due to the presence of the corpus callosum, which acts as a bridge for communication between these cognitive systems. And because of the interaction of these cognitive systems, there is consciousness which arises. Next question. In Sperry's research, which hemisphere is responsible for spatial awareness and visual processing? Again, we discussed that the right hemisphere is responsible for the visual and spatial processing aspects of, uh, visual and spatial processing aspects. So the correct answer is the right hemisphere. So you need to understand one thing. Like the professor also mentioned in the lecture videos, it is not that there is complete lack of processing. It is just that one of them is more dominant than the other. So it's not that the left hemisphere is completely incapable of processing processing these, not that the right hemisphere is completely incapable of processing uh, language and logic, for example, but it is just that they are more dominant in processing these particular processes. Final question. What did Sperry's experiments reveal about communication between the hemispheres in split brain patients? So, what did they reveal? They revealed that in these split brain patients, what happened? The hemispheres were actually functioning independently without any direct communication, right? Because what happened here, you saw that those individuals were actually experiencing some problems. Why? Because they were not having any direct communication with each other. So they did often exhibit conflicting actions because the left and the right hemispheres could not communicate with each other. So in the split brain patients, there was no direct communication and the hemispheres had to function independently from each other. The hemispheres were not able to communicate normally. The left hemisphere was not controlling both sides of the body. That is incorrect. The right hemisphere could does not process the speech and language. So the right hemisphere cannot verbalize stimuli from the left visual field. So that is also incorrect. So the correct option is that the hemispheres functioned independently without direct communication. So, with that, I come to the end of the session. Yeah. So, with that, I come to the end of the session. Uh, in case no one has any questions, I shall close the meeting now. In case you have any questions, feel free to drop them. Thank you, Kushi. Feel free to drop them in the discussion forum. I'd be happy to help you. Thank you very much. Have a happy weekend.